a 0.5 to 5 millisecond delay between the arrival of the action potential at the postsynaptic axon terminal and an actual response taking place on the following cell. So the delay here is due to the time that it takes for the vesicle to dock onto the axon terminal and the release of that neurotransmitter. The delay is not related to the diffusion of the neurotransmitter, right? So that's a common misconception that we want to clarify. So it doesn't take any time for that neurotransmitter to really diffuse across the cleft. Instead, the delay that we see is due to the fact that the axon, the synaptic vesicle must fuse with the membrane here, right? The membrane of this vesicle must fuse with the membrane of the axon terminal, and then the neurotransmitter must be released, and that is the cause of the 0.5 to 5 millisecond delay we see. Alrighty, very important. Let's move on to our third objective here, which is to compare that fast and slow response that I mentioned. And this is based upon two different types of receptors that we see on the postsynaptic cell. So the first type of receptor we see is called a channel-linked receptor, also called an ionotropic receptor. This is an example of a fast response because of the nature of this receptor and the way that it is bringing about a cellular response in this instance. So basically, it's a ligand-gated channel. And just to remind ourselves, a ligand-gated channel needs to be bound by some type of molecule. In this case, it's going to be that specific neurotransmitter that is crossing the cleft and binding onto this receptor. This brings about a rapid or fast change in the membrane potential by the opening up of the channel itself. So in this case, this receptor is also a channel. And so this is a fast response because of that. All right, we don't have to send any signals to any additional structures. When that ligand binds to this receptor, it is also a channel. So we simply have the opening up of that channel and then the movement of ions, which bring about a membrane potential change. As soon as this signal is ceased via the clearing of this neurotransmitter that we just spoke about, then this channel will close and we no longer have the movement of the ions. And so the membrane potential goes back to rest. Okay? So that is the channel-linked receptor or the ionotropic receptor. And that's a fast response. Alternatively, we have G-protein coupled receptors. These are a bit more complex. They're often called metabotropic receptors. And these are much more slow in bringing about a cellular response on our postsynaptic side. Okay, so because of the sheer number of players that are involved in this signal, this is going to take much more time, and that's why this is a slow response. Also, we see an amplification of the signal. So we have the binding of our neurotransmitter to the metabotropic receptor, and as we indicated earlier, this is a G-protein coupled receptor. So the receptor itself is coupled to a G-protein, which is located here. And this G-protein has three different subunits, an alpha, a beta, and a gamma subunit. And the binding of this neurotransmitter triggers the dissociating off of a part of this G-protein here. The alpha subunit floats away and it goes downstream and activates another player in this sequence of events, this enzyme here. We'll talk about some of the names of these structures as we go on, but for right now, we're speaking in very general terms about an enzyme that will then create some type of second messenger, okay? That second messenger will then open up a channel or bring about some other type of cellular response, which is what we want on that second cell. So let's go over that sequence of events again. We've got the binding of that neurotransmitter to this metabotropic receptor. We've got this G-protein that's coupled to this receptor, changing its shape, and a part of that G-protein dissociates off, goes downstream in the membrane, and activates another enzyme that's present in the membrane. The activation of this enzyme will then produce some type of second messenger. We'll talk about different options for second messenger. That second messenger will then bring about the opening of a channel in the membrane, as well as other forms of cellular response. So this can be increasing the calcium production. This can be some other enzymes that are produced that we'll speak about. But basically, the second messenger goes and does several things in the cell that brings about the cellular response that we're looking for. Another form of this metabotropic response is if we have the subunit dissociating off, and so we don't have second messenger here, but the alpha subunit itself goes and opens up or binds to this channel and brings about a cellular response this way. So we can have this G protein receptor bringing about second messenger that opens the channel, or the G protein receptor directly opening up the channel. Either way, these two are examples of slow responses that take some time because we have other players that are involved in signaling this response. Okay. Now, on that basis, we can speak about different types of graded potential. So when that ligand binds to the ion channel, you can open different types of ion channels, and that will bring about different responses on the cell. If we open a sodium or a calcium channel, this is going to be a depolarization in terms of the membrane potential change that we see, and that is called an excitatory postsynaptic potential, or an EPSP, okay? And the reason that this is excitatory or a depolarizing event is because we've got sodium and calcium, both are positively charged ions, both are at higher concentration outside the cell. And so if we open up channels for these ions, we're going to see these positively charged ions rushing into the cell, which bring about a positive membrane potential change. Alternatively, we can open up a potassium channel or a chloride channel, and that would be a hyperpolarizing event, which will bring about an inhibitory postsynaptic potential, or an IPSP. And if we can sort of describe how this changes the membrane potential, we know that potassium has an electrochemical driving force out of the cell. And so if we open up potassium channels, all of this positive charge leaves the cell. So it leaves behind a negative environment, which is a hyperpolarizing event. And then we know that chloride is being moved into the cell. Chloride is negatively charged, and so that excess negative charge moving into the cell is also a hyperpolarizing event, okay? So based upon the charge of the ion and based upon the electrochemical driving force of the ion, we can have either a depolarizing event when we open up that channel, or a hyperpolarizing event when we open up that channel. Now, it's really important, we can also think of this in the reverse, okay? So opening up a sodium or calcium channel would be a depolarizing event, but closing a sodium or calcium channel would be a hyperpolarizing event, okay? Likewise, closing a potassium or chloride channel would be the reverse. It'll be a depolarizing event, okay? So we can think about these as opening the channel and then the reverse stimulus if we close the corresponding channel. So based upon those directions of change of the membrane potential, we have EPSPs and IPSPs. Now we can talk about some different neurotransmitters that are associated with these types of membrane potential change. So EPSPs are often associated with glutamate as the neurotransmitter. And another way to think of this is that if glutamate is the neurotransmitter at the synapse, I will likely open a sodium or a calcium channel or close a potassium or chloride channel, okay? Likewise, if GABA is the neurotransmitter at the synapse, I am either going to close a sodium or calcium channel, or I can open a potassium or chloride channel, okay? So we can do different types of, we can bring about different types of responses by opening or closing the different ion channels. Now, an excitatory synaptic event is going to be more likely to produce an action potential simply because it's going to get us closer to threshold, right? It's going to get us closer to that -55 membrane potential, which is our all or nothing threshold to fire in an action potential. So if we got a positive membrane potential change, we're more likely to get an action potential. Alternatively, an inhibitory postsynaptic event or an inhibitory synaptic communication will take us away from an action potential because it's going to make it harder to fire an action potential because we're now making the membrane potential more negative. It can also be involved in stabilizing the membrane potential, specifically the movement of chloride. We're going to speak about an example where chloride can be used, opening chloride channels, to bring the membrane potential back to rest and to negate any type of positive membrane potential change. So on this slide, we can kind of compare the fast response on the left and the slow response on the right, and the way that the membrane potentials are being influenced here. So we talked about the movement of sodium and potassium through ion channels. And if that channel is a sodium or potassium channel, then we can open it up easily by binding this neurotransmitter to that channel. And we said that the receptor here is acting as the channel as well. So this is going to be a fast response because of that, because the receptor is also the channel. On the right, we can see a much more complex series of events, right? This G protein associated with this metabotropic receptor, and the enzyme that's involved, some type of second messenger involved, and then we open up a channel. So the opening up of the channel in question is way downstream in terms of the series of events, whereas in a fast response, the channel is also the receptor, so it's opened up immediately upon the binding of that neurotransmitter. Okay. Okay, so let's walk through a couple of examples of these events here. So we've got a neurotransmitter binding to a receptor. Now this is a receptor that happens to be for potassium. And so what we see is the opening of this potassium channel. Potassium moves out of the cell, and that leaves behind a negative environment in the cell. So this is an inhibitory synaptic potential, right, an IPSP. On the right, we can see the movement of chloride. So if chloride is moving, again, out of the cell, that is going to negate any positive changes in the membrane potential. Okay? So let's look at an example of how chloride can stabilize the membrane potential. So we can look at this cell on the left, which is no active transport of chloride. So we don't have any chloride channels here. And so chloride is at equilibrium, basically. It's leaking in and out. But on a cell that has a chloride channel that's associated with this ligand, as well as a sodium channel that's also associated with the ligand, for every positive charge that comes into the cell, a negatively charged chloride also comes into the cell. And so what this does is it completely cancels out any membrane potential change. And we can see that here on our voltmeter. There's no membrane potential change because every time a sodium comes in, a chloride comes in, right? Every sodium that comes in, a chloride comes in. And in this way, opening up a chloride channel can act as a stabilizing event of the membrane potential. So preventing any type of depolarizing or hyperpolarizing events of the membrane potential, changes of the membrane potential. All right, any questions so far? Any questions so far? Okay, so let's describe IPSPs here a little bit. So they're basically taking us away from an action potential. So a higher frequency of action potentials can stimulate more neurotransmitter to be released at the synaptic cleft. More neurotransmitter will cross the cleft and will bind to a channel, which will either open or close that channel. And then the greater or lesser ion permeability is what will then influence the movement of ions. And we know the movement of ions will lead to a greater hyperpolarization, okay? And we really want to stress here that hyperpolarization means it's going to be harder to fire an action potential under these circumstances.